Friends, welcome to Biology Made Easy. Today in this video, we'll discuss the chromosome theory of inheritance. So, as all of you know, that Mendel, after working for 7 years, he published all his formulations in the scientific journal in 1866, and but his work remained unknown to most of the scientists in the world because of several reasons. Now, one reason is at that time the communication was very difficult. Just like nowadays, so many social media are there, so many communication methods are there. At that time, in 1866, when he published his work, there was no such communication. That is why his work could not be known to most of the scientists. Number two, he told that the factors which nowadays we are calling genes, they are stable discrete units. This was not acceptable to most of the scientists. Number three, there is no physical proof that actually factors exist. And in addition to all of these things, the most important thing is Mendel applied mathematics and statistical analysis to the biological studies, and it was ahead of time. The scientists were not able to understand why he applied mathematics and statistics to biology. That is why it remained unknown to most of the scientists. But in 1900, three different scientists from different parts of the world, like Hugo de Vries, Correns, and Cermak, they rediscovered Mendelism. Then only after 16 years of his death, because he died in 1884, after 16 years of his death, or you can tell that after 35 years of his work came to limelight. So, and by that time, so many other scientific developments have taken place. By that time, microscope has been developed, and scientists have been able to see the different types of chromosomes. So when all these things came, then two scientists, Sutton and Boveri, in 1902, they put forth one theory that is known as chromosome theory of inheritance. Chromosome theory of inheritance. And particularly in 1903, Sutton, he published a paper in a scientific journal where he directly wrote that the Mendelian factors lies in the chromosomes. Then in later on, other scientists like T.H. Morgan, Sturtevant, and Bridges, they elaborated the chromosome theory of inheritance. They supported it, they elaborated it, they expanded it, that actually the characters, they lie in the chromosomes. So what are the different features that we have to believe that actually the chromosomes are the bearer of hereditary characters? So let us discuss some of the salient features of chromosome theory of inheritance. Now, number one, that the genes are located on chromosomes. This is the first thing. That means all genes physically they are present in the chromosomes. The second, again, important point is genes are present at a specific location on the chromosomes, and that is known as locus. That is again important. Then the next thing, by that time, around 1902 and 1993, the microscope was developed, and scientists were able to identify the chromosomes. That means the chromosomes are located inside the nucleus, and the nucleus it is the controller of all activities. That was known. Then the next thing, in a diploid cell, the number of chromosomes is double. The number of chromosomes is double, and also the number of genes double. That means when the number of chromosomes is doubling, the number of genes is also doubling. Then just the reverse one. In gametes, haploid number of chromosomes are there. That is only one set. Haploid number of chromosomes are present, and hence one set of genes. And the next important point. Now in a diploid cell, the number of chromosomes, the number of genes are double, and in gametes, all are half. Now again, when there will be fertilization, the original characters are restored. In fertilization, the diploid nature of chromosomes, is restored, and the genes become double. Now, in addition to all these things, there are some other important points. What is that? The next important point is there is a parallel behavior between the chromosomes and the genes during meiosis. That is one of the very important points. The behavior of chromosomes and genes is parallel during meiosis. This is a very also important point. And in addition to this, another two important points are there. That is, the next important point is the chromosomes follow Mendel's law of segregation. This is one of the important points. And the other point is chromosomes follow Mendel's law of independent assortment. Because Mendel told that the factors, they are segregating, they are separating, and the factors, they also independently they are asserting themselves. In the same way, the chromosomes are behaving. So let us explain these 2 points. How they follow the law of Mendel's law of segregation and the law of independent assortment. So we have to explain these 2 points. So first we will discuss how the chromosomes they behave, they obey the law of segregation of Mendel. That first we have to discuss. So we are discussing the law of segregation, how the chromosomes are behaving. Suppose at a G1 phase, there are only 4 chromosomes. That means 2n is equal to 4. Suppose. That means what will happen? In this case, there are 4 chromosomes, So these are one homologous pair, and suppose these are another homologous pair. Now in S phase there will be duplication of chromosomes. So in G2, what will happen? In G2 phase, now the chromosomes will be double. The chromosomes are double. Now in anaphase 1, because meiosis is taking place, then meiosis 1 is there, meiosis 2 is there. Now in anaphase 1, what will happen? Now the homologous chromosome will separate two opposite poles, these two also will separate two opposite poles. That means in case of anaphase 1, the two homologous pairs, they will move to opposite poles, and the other pair, they also will move to opposite poles. And at this stage, all the chromosomes are bivalents. But what is happening during anaphase 2? So we will discuss the next page, anaphase 2. In anaphase 2, now this chromosome will divide and the chromatids will move to opposite side, and here, this also the same thing will happen. That means now these two will move to other sides, and these two they will move to other sides. Similarly, this one will move to opposite sides, and these two also will move to opposite sides. Because it is anaphase 2, that means the chromatids are separating. Now the chromatids separating. Now at the end of telophase 2, what will happen? At the end of telophase 2, this will give rise to two gametes, and this will also give rise to two gametes. So it will give rise to now you see now four gametes are produced. So that means the characters which are present in the chromosomes, they are separating, and they are obeying the law of segregation. Now let us discuss this next one, that is how the chromosomes are obeying Mendel's law of independent assortment. So when we discuss that one, now this is more interesting. Suppose now we'll discuss the law of independent assortment. Law of independent assortment, how the chromosomes are behaving at this stage. Suppose there are two chromosomes, and the other homologous pair, other pair is suppose this one. Suppose in one chromosome, suppose here capital R is there, this small r, that means this is a round and wrinkled. And in this case, suppose the color yellow, and here white is there. Now this is one chance. That means in case of metaphase 1, the capital R and capital Y, they are present to one side, and the small r, small y, they are present to the other side. Now when separation will take place, these two will move to one side, these two will move to the other side. So this is probability 1. Probability 1. Now if we will discuss the probability 2, now there is another way is there. Now here the dominant gene capital R is there and small r is there. But suppose here, now this side will be small y and this side will be capital Y. You see, both capital R and capital Y, they were moving to one side. But in this case, capital R and small y, they are moving to one side, and the small r, and that means say wrinkle and yellow, they are going to other side. That means see here round and yellow, they are moving to one side, but here round with the white color, they are moving to other side. This is independent assortment. So if this is the arrangement at metaphase stage, then naturally in anaphase, what will happen? In case of anaphase, now this is anaphase 1. This is anaphase 1 or meiosis 1. That means say if this is capital R, this is small r, and if this one, then it may be small y, it may be capital Y. And in this case you will find just the reverse one. In this case you will find that capital R and this is small r, while this one, this is capital R, this is small r, this is capital Y, this is small y. That means here the arrangement is like this, but here the arrangement is like this. So ultimately in anaphase 2 what will happen? Now in this case, now this will move to one side, this will move to one side, this will move to one side, this will move to one side. While in this case, you see, this is capital R, this is capital R, while these are small r, small r. Here this is capital Y, here small y and small y. But here in this case you see what is happening. In this case, this will be the arrangement. That means this is capital R, this is capital R, this is small r, this is small r. But what is the arrangement in this case of Y gene? This is small y, this is small y, while this is capital Y, this is capital Y. So this is anaphase 2. This is anaphase 2. Now what will happen at the end of telophase 2? At the end of telophase 2, it will give rise to gametes like this, and this will also give it. That means a round is associated with the white color and a wrinkle is associated with the yellow color. But here a round is associated with yellow color and wrinkle is associated with it. That means say all the 4 types of gametes can be formed due to independent assortment. Now if I will give another example, very simple example, it will be still better you can understand. Suppose let us take 3 genes. Suppose the 3 genes are, this is A, B, C. So naturally the alleles will be A, B, C. Suppose in metaphase, all the dominant alleles are present towards one side and the recessive alleles are to the other side. So in this case, how many types of gametes can be produced? Only 2 types. Now let us see this. What is the other example? Now suppose capital A, capital B will be one side and this small c will be one side, and this side small a, small b, but capital C. If they will move to their opposite poles, now these also will give rise to 2 different types of gametes. It depends simply independent assortment. Now what is the other chance? Now suppose here you will take capital A, small b, capital C. Then it will be small a, capital B and small c. Then again we see here A, B both dominant are going to one side. but here ASC are going to other side while capital B is going to other side. Now you see the next one. The next one will be small a, capital B, capital C, capital A, small b, small c. That means these are the different, when three genes are there, we are considering these are the different probabilities. This will give rise to two types of gametes. This will give rise to two types. This will give rise to two types. This will give rise to two types. That means altogether 8 types of gametes can be produced. And you know the formula that 2 to the power n, and here n is equal to the number of the alleles. So naturally it will be 2 to the power 3 is equal to 8. So you see here 2 types, 2 types, 2 types, 2 types, 8 types are there. And if you consider the case of human beings, in case of human beings, there are the 2n is equal to 46, that means n is equal to 23. If there is no crossing over, then what is the probability that independent assortment will take place? So naturally it will be 2 to the power 23, that means it will come to millions. That means if there is no crossing over, the chance, the probability of arrangement of the different types of chromosomes in a human being will come to millions. So in this way, the chromosomes also, they obey Mendel's law of independent assortment, also law of segregation. So now it is clear how the chromosomes, they are the bearers of heredity or they are the carriers of heredity. Now let us discuss MCQs. So one MCQ, the first one, Sutton and Boveri noted that the behavior of chromosomes is parallel to the behavior of first choice genes, second choice chromatids, third choice nucleus, and fourth choice nucleolus. This is one question. Now come to the second question. In 1902, Sutton and Boveri formulated the law of one choice chromosome theory of linkage, second one, chromosome theory of inheritance, third choice, coupling and repulsion, and the fourth choice is the theory of linear arrangement of genes. So these are the type of questions usually asked from chromosome theory of inheritance. I hope now we have a very clear understanding of chromosome theory of inheritance. Thank you very much. God bless you. Have a good day.